Thank you, Mark, for the introduction. So what I will try to do is just give you some kind of a snapshot of a lot of things that are actually going on the field of gut health diagnostics and gut health biomarkers. It's actually a quite complicated topic, and you can use a lot of different technologies to actually identify good gut health biomarkers. So the first thing, I will just pave the way a bit for other speakers that will follow my talk. For example, one of the postdocs of our group is presenting really a practical solution in a few presentations. But also in this presentation, I will just try to give you an overview of which potential biomarkers can be of value and which are the ones that are currently being developed. So I'm quite confident that within the next few years, some tools will be available to be used on farms, on flocks, to assess or to measure gut health. First question, of course, is what is gut health? It's a very complicated thing. There are a lot of different definitions. And of course, we first have to have an idea on what we mean with intestinal health before we can, of course, try to identify and quantify this kind of markers. Now, I'm not going to talk about humans, but just there's a lot of interaction with a lot of knowledge present in human medical science that we can actually use. And I just want to give you 2 slides on the current diagnostic tools in humans to measure intestinal health or intestinal inflammation. And of course, you don't go to a medical doctor and give him a sample and ask him, okay, please, what is my gut health status? That's something you don't do. So there's nothing available there, of course, to measure general intestinal health status. Of course, when you have an infection, then, of course, they take a sample, they just plate it onto agars, and they will tell you which pathogen you're infected with. That's, of course, also the case for poultry. But there's one well-validated method that they use in clinics to actually measure intestinal inflammation, and that is based on one specific protein, which is calprotectin, and calprotectin is actually a marker for intestinal inflammation in humans. And what this protein is, in fact, it's a protein that is present within the granules, the secretory granules of granulocytes, of neutrophils. What you see here, for example, on the left, this is just an intestinal villus. You see these immune cells, these granulocytes that really infiltrate the gut wall when there's some kind of a trigger. Then they translocate through the epithelium and they end up in the intestinal lumen. And also they degranulate. What you see here, they release granules, they release proteins, and these proteins, they end up in the feces, okay? And actually you can measure quantities of this specific protein in the feces. And that's also something that would, of course, be of value for poultry. And I will come back to this at the end stage of my talk, because there are similar proteins that can as well be used in chickens. What you see on the right side here is just a graph showing the quantification of this calprotectin in different human subsets. On the left, you see Crohn's disease patients, then UC, that is ulcerative colitis, IBS is irritable bowel syndrome, and then you have these healthy controls. I hope this is a bit visible, okay. So, but what you can see here is that in cases of severe inflammation, you have very high levels. But in cases of, well, there's something not normal, a bit of diarrhea, maybe a bit of constipation, just some limited problem, limited inflammation, you don't see any difference between this population and the control population. So actually, you need really severe inflammation to be able to measure it. And this is, of course, kind of a problem. And especially when you look into poultry, you also have this, on the one hand, cases of severe inflammatory diseases. Necrotic enteritis is probably the most well-known. You have a massive inflammation of granulocytes there, and also this kind of proteins will actually be released in intestinal lumen, and you can measure these in the feces. But on the right side, you see, well, this is just subtle problems. Let's say in chicken terms, in broiler terms, that means a little bit of performance loss because of a little bit of intestinal inflammation, some dysbiosis, and so on. And that's a complexity, because for this kind of condition, there's not, well, it's maybe not severe enough to find this kind of biomarkers that are used to measure severe inflammation. But I will come back to this in a few minutes, because actually there are some possibilities there. So this kind of problem, just as a introduction, this kind of, well, subtle intestinal inflammation or dysbiosis, they can be evoked by a lot of different triggers. Can be mycotoxins, can be clostridial toxins, can be the diet composition is very important, and so on. There are a lot of potential factors that influence this kind of conditions, okay? So there are a lot of different potential triggers. So we are not talking about measuring the triggers, but measuring, of course, the outcome of this. I will come back to this again. Now, what we are really looking into is we want to try to identify some predictive biomarkers, and that's a challenge, okay? What we try to do is, like in broilers, taking samples at about 2 weeks of age, measuring some kind of a biomarker, and then predict what is going to happen in the near future, what the performance will be, whether there will be health problems or not, and so on. So that would be the ideal biomarker, something that is predictive for performance. Now, oops. I just told you I will give you some definitions. This is something you already know, of course. Macroscopically, when you have the animal has intestinal health problems, you will just see it. Of course, in cases of severe disease, necrotic enteritis, you see gut necrosis, but also there's a lot of other things you can see when you open the bird, you look at the macroscopical appearance of the gut wall, okay? And there are a lot of people that have been using or implementing this gut health scoring systems, looking at the gut and just giving a value there to be used. Of course, this is kind of, well, it's not completely objective, I would say. It is a little bit subjective, depends on the person that's doing that. Here's one of them. Just looking at different entities, looking at thickness of the gut wall, at inflammation, undigested feed particles, and so on. And this at least gives you an indication or gives you a score at the end about, well, intestinal health. But as said, it should be ideal to replace this kind of system with a very easy tool. Take a fecal sample, put it in a machine, get the value. That's basically what we are looking for. So now, you can also look at it under the microscope, of course, and that gives you a lot of information of what is actually going on. And if you look under the microscope of animals, of which the veterinarians are saying, here's a problem, this is dysbiosis or wet litter or whatever, then you will see that villus length is decreased, you have some intestinal inflammation, you have a lot of immune cells that infiltrate the gut wall. So this is something that can learn you a lot about what is actually going on and what you need to measure, okay? So just some pictures here. This is on the left side necrotic enteritis, okay? This is the most severe case of dysbiosis, which is actually gut necrosis, and you see that here, the whole structure of the gut wall is completely disrupted. This is normal on the right side, normal gut wall appearance and also long, slender villi. And in the middle, this is just some congested blood vessels, an inflamed gut. And what you can see here is the villi are thicker, a bit blunted, they're shorter. And all the brown dots, these are immune cells, okay? In this case, these are CD3 positive T cells, so these are T lymphocytes. So that actually means that in this middle case, in which the animals don't have any symptom, you don't see anything on the animals. The only thing that they will do is perform a little bit less because they have to spend a lot of energy in this immune cell infiltration. It costs a lot of energy that the animals are not putting in muscle, of course, that's quite evident. So actually, just a little bit of a short molecular explanation of what is actually going on. And if you have gut inflammation, that basically means that there will be some disturbance in intestinal integrity. So this is actually the normal situation. All these dots here, these are bacteria with different colors, different genera, different species, different families, whatever. And actually, what is happening is that in a normal individual, also us here, but also normal chicken that is perfectly healthy, they just do not react to all these bacteria that are sitting in the gut lumen. We are tolerant against these. And actually, when there's some kind of a breach here, when you have epithelial cells that are dying due to toxins or mycotoxins or even coccidiosis, you will have some of these bacteria that go through these lesions and connect with receptors that are basically at the basolateral surface of these epithelial cells, and that will be a trigger for gut inflammation. That's what you can see here. What you see here, this is an increased permeability, so some gut damage, some damage to the tight junctions. That's what you see here in the middle. Cells not connecting very closely, and there's a breach, and then you have these bacteria, toxins, maybe dietary antigens that leak through. They connect with some receptors here, and then, of course, you have cytokine expression and inflammation. That's basically what we need to find some kind of biomarkers for. If you look well here, you also see a lot of these red bacteria, so you have also dysbiosis quite often. Already mentioned by Joël, if you have inflammation, you see a microbial shift, and you see some bacterial populations going up, others going down. That's also something very specific. Now, at the end, you will also see that, as I just mentioned, granulocytes go through to the intestinal lumen. It doesn't really look like a granulocyte. I tried to draw it, sorry. And all these blue things here, these are just also proteins from serum, whatever, that also are ending up in the feces, coming from the bloodstream, from the intestinal mucosa, and this just ends up in fecal material. And actually, what I want to point out is that if you look for biomarkers, if you want to identify biomarkers, you can look at microbial populations, microbial shifts, microbial functionalities that are different. You can look at these host molecules that are leaking through and end up in fecal material. You can look at inflammation markers, at cell death markers, and so on, okay? So this is something that you can use a lot of different technologies for to try to identify these different proteins or molecules. So I think this is an important one. There's a lot of novel technologies being used for the moment. Joël talked about metagenomics. You also have metaproteomics, metametabolomics, and so on. There's a huge set of novel techniques that you can use and really use on experimental models, inflammation models, and try to identify all these different potential biomarkers, and that's actually what we are trying to do. And you can learn a lot from that, and especially you can identify potential markers. So first of all, microbial shifts. If you think about microbial shifts, you think about 16S sequences. This is differences in families, genera, species. You also think about different functionality, meaning functional genes in this microbiome will be changing. You also think about metabolites, bacterial metabolites. Bacteria produce a lot of different molecules and metabolites, and also when you have some kind of dysbiosis, different microbial composition, actually the metabolic profile of this microbiota is changing. And this is also something you can measure, okay? Question is, of course, which metabolites are linked with dysbiosis and inflammation, and which ones we should actually measure. Now, I will come back to that and give a reply to this in a few slides. So, just a third definition of gut health, okay? And this is a microbiological definition saying that if the microbiological diversity is high, this really is related to intestinal health. And of course, it should be a microbial composition that produces metabolites that are basically anti-inflammatory and not pro-inflammatory. That's a very vague definition. I will come back to this in a few slides. In fact, already Joël talked a lot about it, because when you look at the human data sets, you can see which bacteria are doing what. You have the butyrate producers, this is anti-inflammatory. You have some others that are pro-inflammatory. And of course, also these you can try to use as biomarkers. This is already mentioned by Joël. We have also been using this kind of model. It's a different experiment, but to identify 16S sequences that are actually either or not associated with performance and with inflammation. What you see here is it's just a model, okay? It's a little bit unpractical, but what we wanted to do here is induce intestinal inflammation, induce villus length decreases, induce performance losses, induce some kind of a pathology between brackets that is more or less identical as what you sometimes see in birds that do not perform well. So actually what we have been doing is using polydigestible diet, no enzymes. We depleted microbial composition using antibiotics. Then, in fact, a bacterial cocktail was given of some opportunistic pathogens, E. coli, some non-pathogenic Clostridia, even some lactobacilli. And then also we gave a coccidiosis challenge, okay? And then we just sampled and looked at 16S sequencing. So basically the question is, first of all, is this a good model? Secondly, what type of microbial populations are changing, okay? These are some data. Has already been mentioned, but what you see here on the left is that, of course, in the challenged animals, you have a body weight decrease, you have a feed conversion increase. That's evident, so the animals are clearly performing less. You see some coccidiosis lesions on the right side, so each dot here is one animal, and these are the animals at day 26, so more or less 3 to 4 weeks of age. This is a dysbiosis score. This is just a macroscopical gut wall appearance score, the ones I just showed you some pictures of, okay? This is also increased. So you see some gut wall abnormalities if you open the animal and look at the intestine, and you also see performance losses. And if you look to the histology, you also see villus length decreases and inflammation. Just showing you this data to tell you that this is a model that you can use to identify potential biomarkers. That's basically what I'm saying here, okay? What you see here on the left, this is the villus length decrease. It goes from over 2,000 to 2,500 micrometer in the duodenum, which is quite strong. It's a 25% reduction in villus length. Villi are also thicker, and as you can see here, right below, this is a picture of the challenged animals, meaning there's a massive immune cell infiltration. These are T lymphocytes again. I also see that on the graph, about 15% of the whole gut mucosa is filled with this brown staining, and this brown staining are brown cells, and these are T lymphocytes, okay? Now, if you do 16S sequencing, you immediately see that the challenged animals, they have a lower diversity, okay? That's one thing. But importantly as well is that if you compare control animals with challenged animals, and you look at the observed taxonomical units, so numbers of bacterial taxa, if you want. In the control population, it's evident, well, animals that weigh a little bit less or more, they all have the same diversity. But in challenged animals, you see this very clear correlation, meaning that animals under challenge, and this has already been mentioned by Joël in the previous talk, animals under challenge with a high diversity, they have a higher body weight. They have less problems of coping with the challenge, okay? Animals with a lower bacterial diversity seem to, well, be the ones that react very strong to the challenge. This is also linked with immune cell infiltration and so on. You can clearly link that with bacterial diversity, and this is something you can measure. It's still a bit of complex to measure it, but anyway, this is in principle a tool to look at responses to challenges. So, going much more into detail, in fact, we have a full list of all sequences that are going up and down and so on. But what always comes back in this kind of models and also in other inflammation models is that quite a lot of bacteria that are depleted in challenged conditions, these are butyrate formers, okay? What you can see here, these are, in blue, the families of Ruminococcaceae, Lachnospiraceae, which are broad families that contain a lot of butyric acid producers. And in a lot of different models, also in mice, even in pigs, but also in chickens under different conditions, if you challenge the animals, also with coccidiosis, also with some other pathogens, you see a depletion in these specific families. And there are a lot of other names there. I just highlighted two of them as well. Fecalibacterium porosnitzii, the one that Joël mentioned as being very, very strongly connected with intestinal health in humans. We also find it here as like 1,000-fold depleted in challenged animals. Butyriciococcus is another one. This is one that we have been isolating from chickens before, and as if you administer this one to the chickens, actually they improve their intestinal health. And also here you see this huge depletion. What you see here on this graph in green, that is the, well, what is more abundant in control animals, but actually it's not more abundant in control animals, it's depleted when you challenge the animals. That's basically what it says. So potentially, if you just quantify these populations, you have something to work with, okay? And quantifying these populations is fairly easy. You can just use quantitative PCR methods, apply it to fecal samples or to intestinal samples, and actually measure abundances of these populations, which is fairly easy to do. Okay, just One step back to humans, just as I just mentioned, this is something that looks very similar to human intestinal inflammation. What you see here is just a study comparing human healthy individuals with people with inflammatory bowel disease. And actually, in people with inflammatory bowel disease, you see a depletion of butyrate producers, Lachnospiraceae family, identical as what I'm just showing. An increase in proteobacteria, so this contains a lot of Enterobacteriaceae, a lot of E. coli, and opportunistic pathogenic organisms. You also see an increase in Clostridium perfringens, for example. So there's like a balance, okay? On the one hand, you have the strictly anaerobic butyrate producers and all the other ones that are connected with it, and on the other hand, you have these Enterobacteriaceae, Clostridium perfringens, and some others. And it's really about the balance between these two populations. It is also easy to quantify. Again, what they have been found in this paper also is that Butyriciococcus, for example, is actually depleted quite severely in ulcerative colitis patients, and Escherichia coli, of course, is increased in abundance in these disease entities. So it's very similar. And it seems to be that if you look at microbial composition over different animal species, also humans, there's a lot of similarities in the way microbes respond to inflammation, to challenge, and the way that the host is reacting to microbial disturbances. Okay, just to show you that, well, in this paper we have been working together with people from human medical departments in the University of Leuven. In fact, people that develop severe inflammation after resection of parts of the gut in inflammatory bowel disease patients, the outcome of what happens there is really depending on microbial composition, and people with high concentrations of Clostridium perfringens, they will develop severe inflammation leading to an additional surgery. So Clostridium perfringens clearly linked to severe pathology here. Also some papers showing the Enterobacteriaceae and proteobacteria that are very important here, and also many other papers showing that butyrate formers are very important also in human gut inflammation. And this is one that we have been publishing a few years ago in Gut, in the human science paper. So showing that, in fact, a strain or genus that we have been isolating from chickens is actually also found in humans, and this is also very important there in gut inflammation. So the less abundant this bacterium is, the more chance that people get gut health problems. Okay. And you can do exactly the same with the carotid enteritis. You see similar shifts. So it's all a bit related. Now, of course, butyrate formers are important. I'm not going to focus too much on it, but that means, of course, well, it's butyrate production, and butyrate is a very strong anti-inflammatory compound. It strengthens epithelial cell layer integrity. It increases expression of tight junctions. It's causing cell proliferation and differentiation, so it's a very important compound. So also here, measuring butyrate concentrations could actually be of relevance, especially when you also start measuring other fatty acid profiles. That's also something that can potentially be done. Now, one of the postdocs in our laboratory has looked at all these 16S sequences and then just tried to map functional pathways. We're just wondering at that moment what is really happening in a functional way. And if you do that, it's a little bit of a complex picture, but actually what I want to point out here is that challenged animals, chickens, under the gut inflammation models, they actually have a depletion in butyrate production pathways, and they have an increase in ammoniac and hydrogen sulfate producing bacterial communities. And this is quite important as well, because in fact, this means that you can look at, let's say, quantifying genes that encode functional pathways, just using quantitative PCR, and that would also give you some gut health biomarkers. I will come back to this in a few minutes. It's about if you would be able to measure the potential of the bacteria to produce butyrate, the potential to produce hydrogen sulfide, to produce ammoniac, and try to bring this in a quantitative way, you actually have a good idea on microbial health or intestinal health, but of course by measuring functional activities of the microbial population. Just to explain this, this is the normal situation, a lot of diversity, anaerobic conditions, of course, because you're speaking about the gut, the intestine, high microbial diversity, and a lot of butyrate producers. This butyrate is taken up. There's It's oxidized in the cells, oxygen is used, and this is used as an energy source. On the other hand, you can have this, well, microbial population shift, loss of diversity, depletion of butyrate producers, and an expansion of these gram-negative proteobacteria, which contain all E. coli-like organisms. And actually, what then happens is that there are some problems in butyrate production. Butyrate is not taken up anymore by epithelial cells. You even have glucose being used by epithelial cells, lactate being produced, and it all becomes a bit more aerobic, because what you get is immune cell infiltration. These are granulocytes. You get reactive oxygen species. You get more or less well, conditions that are not strictly anaerobic anymore. And this type of condition also means that you are killing off strictly anaerobic bacteria even more. So it's like a vicious circle. You're also killing off these strictly anaerobic butyrate producers. So this depletion in this kind of bacteria is even further in expansion. So again, you also have production of nitrates by immune cells. You will have a reduction and production of ammoniac. You will have sulfate-reducing bacteria. Just, in fact, oxygen, but also nitrates, sulfates, used as electron acceptors by bacteria, and production of different components that are toxic, okay? That's basically what this is saying. Healthy conditions, let's challenge conditions. That's what is happening. I think this is now all quite well documented in different animal species, but also in chickens, this seems to occur. And in fact, that means that, as I just mentioned, you can start quantifying these functional pathways. If you make a ratio between this enzyme, this gene, bacterial gene that encodes this enzyme, and you make a ratio between this one that is involved in butyrate production and the other ones, then actually you also have a value that gives you a hint on intestinal microbial health, okay? This is giving you a hint on, okay, is the microbial community in the gut positive, beneficial, or potentially toxic? That's basically what this is meaning. So just to explain a bit more on the mechanistically what actually this means for an epithelial cell. Why is this so important? Well, this is an epithelial cell. You have some transporters. These are transporters for butyrate. This is a monocarboxylic acid transporter, another transporter. You also have receptors. These are receptors for butyrate amongst some other small molecules. And actually, what is happening is that these compounds, such as ammonia, for example, produced under challenged conditions, they block expression of these transporters. Hydrogen sulfide, it's blocking beta oxidation. So again, there's this balance. You have butyrate, then you have a lot of other metabolites that are harmful. So there's always this balance that you have, and you can actually quantify one or the other and make these kind of ratios. Luckily, you also have some other compounds that bind to these butyrate receptors, and the difference is here that these receptors actually, they promote anti-inflammatory activity, okay? So maybe a nice thing, but that's not about diagnostics, maybe about feed additives, is to try to figure out how we can activate these kind of receptors that will stimulate anti-inflammatory properties, okay? But that's something that is not of relevance in this specific talk. I talked a lot about 16S sequencing, functional pathways. Of course, you can also do metabolomics, or metametabolomics even, looking at what type of metabolites are present in the intestinal lumen, or in fecal material. And what you see here, just one specific example. We have a huge data set of all different metabolites that are going up and down when you challenge chickens with coccidiosis or Clostridium or any dietary challenge, for example. And what you often see is that proteolytic activity is going up, meaning you have some products here that are actually a result of protein degradation or protein fermentation. And that's something that's very specific also for a lot of lactic acid bacteria, but also Enterobacteriaceae. So this points to this shift I was just explaining. Cadaverin and putrescine, these are polyamines. These are well known in humans because they are really associated with colorectal cancer there. It's completely irrelevant for chickens, of course, but still. Actually, these are also compounds that are produced by the same populations. So lactic acid bacteria, but also a lot of Enterobacteriaceae. So this is just an example. If you can measure this kind of compounds, you actually also have some kind of markers for intestinal health. So these are all potential targets. So I think using all these meta technologies, you can just identify a lot of potential targets that you can quantify. And then, of course, the major question will be, this all works in experimental models, but you have to apply that to field conditions, okay, and test in the field. And this is something we are currently working on. So final part of the talk is about host responses. So what we now had in this first 25 minutes is microbial biomarkers, and actually there are a lot that you can actually use, and this is something that is currently being developed, so I hope that in a few years there will be something available, but also on the host side, you can have a lot of things. Because if you look at the host, you can look at gut inflammation markers, you can look at epithelial cell markers, because if you have epithelial cell death, it will anyway end up in the feces, okay? All these molecules, dead cells, proteins coming from these dead cells, and also, of course, some proteins that leak out from plasma to the intestinal lumen. And these you can also find in fecal material. But only, of course, if they are not degraded. And that's a major challenge. You have a lot of proteins that are present, host proteins that are like produced and end up in fecal material and that are much more abundant in challenged versus control animals. But of course, there are a lot of proteases as well. A lot of proteases that break down all these different proteins. So you need to find stable proteins that are not broken down by proteases that are found in fecal material. And then you have an ideal biomarker. That's what calprotectin is. Calprotectin is an inflammatory marker in humans that is very stable. And you can actually find that in stool samples. Of course, in chickens, it's a bit more complicated. Because stool sample is easy to give. In chickens, you can take fresh samples, but ideally you should use something on litter. So there's one additional talk later on about this, because these are 3 different models I'm showing here: a coccidiosis model, necrotic enteritis model, and the model I was just explaining. In a few minutes, Evie will explain you about a biomarker that is found to be increased in abundance in all 3 types of conditions. I will leave that for her. But actually what I will do is just describe you a few biomarkers that you can find in this model, okay? And one of them, we did some proteomic analysis, some metaproteomic analysis, trying to find proteins that you find in intestinal content, in colonic content, and in fecal material that are more abundant in challenged animals. And actually, one of them is this. This is myeloid protein 1, and this is in fact found in chicken granulocytes. It's exactly the same type of molecule as calprotectin in humans, but this is a protein that you find in granules of heterophilic granulocytes, so the chicken counterpart of the human neutrophil, and you find that increased in colonic content of animals under challenged conditions, highly increased. So it means that this is a stable compound, it's a stable protein that is present when the animals have some kind of gut damage and gut inflammation, because this is really pointing towards granulocyte infiltration in the gut wall. So this is clearly a nice biomarker, and of course, this kind of molecules you can detect using ELISA and further on, okay? You can quantify them. So just about developing antibodies and setting up a test. So another one, I just give you a few examples. We have a whole list of different proteins that you can actually find to be increased in abundance, either in challenged conditions, but you also have some that tend to be, well, present at high concentrations in control animals and are depleted in challenged animals, okay? So you have like a variety of different proteins actually being present. This is one, it's called aminopeptidase EY. It's just involved in protein digestion, okay? And if you have food proteins, of course, they are, first of all, converted to polypeptides in the stomach, but then later on, smaller peptides, they are further broken down to amino acids and then, of course, taken up by epithelial cells. And this aminopeptidase EY, this is just a brush border enzyme. It's produced by epithelial cells. It's present in the brush border to digest proteins. Actually, what it means is that the more epithelial cells you have, the more of this kind of protease or peptidase is being produced. if you have shorter villi, if you have less epithelial cells, you will have less production. That's one thing, or maybe it's just less produced anyway. But actually, in this kind of inflammation models we have been using, you see a serious depletion or decrease in ileal and colonic content, and also in fecal content material. Actually, there's a depression or a decrease in concentration of these kind of proteins in the intestinal content there in challenged animals. So it's some kind of an epithelial cell abundance marker. I have two other examples before I close. This is fibronectin. It's in fact a compound or a molecule that is found in the basal membrane in extracellular matrix. And it also makes sense because you find it much more abundant in colonic content of challenged animals. And of course, that's because you have some kind of gut damage, okay? If you have gut damage, this compound will be released in the intestinal lumen because actually it's part of the basal membrane to which all the epithelial cells are connecting, okay? If epithelial cells are dying, this extracellular matrix components and also basal membrane components will just be released in the intestinal content, and actually you can also measure this. Now, there are quite some other proteins that are all having to do with inflammation, gut damage, even epithelial layer integrity and so on that you can actually find there. And I think this is very promising, okay? Because it's quantifiable in an easy way. And the first thing, of course, is developing antibodies, developing ELISAs. This is something we are currently doing. And we are currently also just using field samples and using these kind of ELISA systems on the field samples to check whether abundance of these proteins is actually connected with animal performance. And if you have that, you have something beautiful, of course, and especially when you would measure it, like, let's say, at 2 or 3 weeks of age, and then predict what is going to happen at later stages. So what next? Well, we know that a lot of these technologies that are currently being used will still generate much more data than I have been presenting here. It's just a snapshot. But ideally, this is the way to go. This is, in fact, a calprotectin assay. It's a bit of pregnancy stick. But you can just dilute a fecal sample, put it here. It's like lateral flow system. And it can contain different markers if you want, okay? If you, for example, put 3 or 4 different protein markers on such a stick, you have quantification of different ones, and above a certain threshold, this will be positive or negative, and gives you an indication on intestinal health to be used to, for example, evaluate feed additives, to be used to evaluate intestinal health of a flock, and so on. I think this is something that can be very important for the future. Just the one but final slide, I just looked into the literature about easy systems to measure this kind of, do this kind of quantification. And actually, you have quite some companies that are already now having systems available which you just put this kind of dipstick in the smartphone and actually read it, okay? You just take a smartphone, either you take a picture of the color or you just put a stick in the smartphone, actually you have the value. This is, I think, very promising, and there's quite a lot of papers in this specific journal, biosensors and bioelectronics. I didn't know it at all, but they describe these smartphone applications, these biosensors. So this is something I think this is clearly what we should do next. Final slide, I want to thank a lot of people. Once, all the people in our group, and especially also, of course, Vanessa doing a lot of work in the animal modeling, also Evie doing a lot of work on bioinformatics, and Fien also doing a lot of work in the animal models itself. And also the proteomics and metabolomics group at our university. And of course also the multiple industrial collaborators we have, of which some of them are on the sponsor list here, and also the people from VetWorks Pulform, because actually if we do animal models and animal trials, we often discuss it with them, and also for analysis of field samples. This is together with Maarten and his companies. So that's basically what I wanted to tell you. Thank you.